So this goes into a bunch of different models about how enzymes work. So early models. So one of the earliest models for enzyme how enzymes work was called the lock and key model. And so this is just thinking. So this is in 1894 by Emil Fischer. And so what this was trying to explain was the substrate specificity of enzymes. So you can think again of this molecular shape of an enzyme. And so what Emil Fischer said was that the enzyme just complements the shape of the substrate, okay? And that helps to explain the substrate specificity, right? And so if you look at this, you just see, okay, here's the shape of the molecule, and like a puzzle piece, it looks like the enzyme fits the substrate, and this allows for the substrate to bind, and then there's some sort of chemistry to form in this diagram two products, okay? So that's a very intuitive way to think about this that helps to explain the substrate specificity. However, it has some problems when we think about the energetics of the enzyme, because let's say we have, so these are just reaction coordinate diagrams. So the hills, again, are transition states, okay, in here, and then we can see these sort of wells at the bottom of the diagram. Okay, so this is just another way to denote a reaction coordinate. And so let's label these. So the first potential well is the substrate for this reaction. So here's S, and then the last potential well is P. Okay. All right, so we're going to follow first this yellow trace. Okay, so this is the uncatalyzed reaction, conversion of S to P. All right, so we can think of there being a transition state. It's just an elementary reaction of S conversion to P, and there's one transition state between it, and we've labeled it here as TS. Okay. Now let's think of the catalyzed reaction, or the enzyme catalyzed reaction, right? And so if an enzyme instead takes up substrate to form an intermediate, which in this case is the enzyme substrate complex, we have a second potential well, right? There's a transition state in between here, and then there's a second transition state between formation of the enzyme substrate complex and formation of product. Okay. What the lock and key model suggests is that because the enzyme is suited particularly to bind to the substrate and evolve to the substrate, then it's going to bind to the substrate with really high affinity. Because remember, all those molecular recognition sites are to be enthalpically favorable contributions to the energy. So what you may actually expect then, by comparing the uncatalyzed versus the catalyzed reaction, is that the intermediate, which is the enzyme substrate complex, is actually going to be more energetically favorable than just the free enzyme plus the substrate. Okay. Now, if we still need to go through the same transition state, which has the same energy, that actually makes it harder to get the activation energy, right? Because if we look at the uncatalyzed reaction, the activation energy is going to be coming from the first potential well to the first transition state, which, using this hypothetical situation here, we're calculating as 30 kilojoules per mole, versus the catalyzed reaction, right, is going to be 35 kilojoules per mole, because we have a larger span of energy between the enzyme substrate complex and this transition state 2, okay, which is analogous to the first transition state. So that's a bit of an issue, right? So the model doesn't really appropriately connect to the idea that enzymes are catalysts and lower activation energy. So in order to consider this, we have to think of a different model. And so Daniel Koshland came up with a different model called the Induced Fit model. Okay, so one way, when the original thought of this was that the enzyme actually strains the substrate to force it into the transition state. And so there may be a distortion of the site, which looks kind of like the substrate, but not perfectly. And then it forces the substrate to take more of the shape of the transition state, which promotes the chemistry. And so another idea that's important in this is the idea of protein conformation changes. So we talked about this with hemoglobin, right? And hemoglobin is able to have conformational dynamics or changes in shapes. And so an important thought on this too is that the enzyme itself might change shape. So first, it has a shape that allows it to bind to the substrate tight, but not too tightly. But then it changes shape in order to accommodate either force the transition state, or as this later got changed, to stabilize the transition state, right? So there may be some shape change between the time the enzyme binds the substrate and when it starts to catalyze the reaction, right? That allows it to instead more appropriately stabilize the shape of the transition state instead of the substrate. Okay, and then if we think about the energy diagram for that, that starts to make a little bit more sense, because then in the substrate, the intermediate, again, which is the enzyme substrate complex, may be a little bit higher energy than just the enzyme plus the substrate itself. So it avoids that problem with the lock and key model, that the enzyme substrate complex is actually more energetically favorable than just the free enzyme plus the substrate, which would cause a problem with the activation energy. But in addition, you also think about stabilizing the transition state, and so that drops the energy of the transition state down. Okay, so again, in our example here, instead of the catalyzed reaction, which has a transition state energy of 30 kilojoules per mole, it drops down to 20. Okay, and so the net effect for the uncatalyzed reaction, again, is 30 kilojoules per mole versus the catalyzed reaction. The activation energy is effectively going to be 20 kilojoules per mole. So that makes more sense in how we think about how enzymes work, and that's to decrease the activation energy, or one mode of how enzymes work is decrease the activation energy in order to increase the rate of the reaction. So again, take-home point here to summarize is the structure of the enzyme stabilizes the transition state. So the molecular interactions, right, are there to stabilize the shape of the transition state, not necessarily the substrate.